So I am going to be talking about polyreactive B cells in autoantibody-positive in patients with new-onset type 1 diabetes. So this talk is actually going to be in some ways fairly similar to Eric. So he has a lot of great introductions for me. So what we are really focused on is type 1 diabetes, and there's a lot of reasons to study type 1 diabetes, but one of the big ones is that the incidence has been increasing globally over time. So for some reason, we're seeing increases in numbers of these type 1 diabetes cases. And additionally, it's a disease that particularly affects young individuals. So almost 50% of new diagnoses are in individuals who are under the age of 20 years old. So once you get diagnosed, this is then, of course, a lifelong disease. And type 1 diabetes is generally caused by beta cell loss. So beta cells are located in the pancreas, specifically within these pieces called islets. And in normal beta cell function, beta cells actually sense glucose from the environment, and in response, they secrete insulin. So this insulin secretion helps decrease blood glucose levels and prevents diabetes. However, in type 1 diabetes, without beta cells present, this glucose can't be sensed. We don't have insulin secretion, and instead we have increased blood glucose, which then leads to all of the type 1 diabetes impacts that we see, including a lot of different organ damage. So in general, we think that beta cells are destroyed by T cells. It's described as a T cell mediated disease. However, it's the whole immune system interacting that really causes this destruction. So both B cells and different myeloid cell populations present antigen to these T cells, which then go on to attack and destroy the beta cell. And then, of course, the beta cell has a role in that it then releases those antigens to then be presented by the myeloid cells and B cells. So it's really this cyclic process. And there's a lot of cell types that are involved in type 1 diabetes, which include the beta cell, as I described, all of the different T cell subsets, all of the different lymphoid populations, and then, of course, also the B cell. So my team is really focused on using a lot of different sequencing technologies to determine the role of all of these different cell types in type 1 and type 2 diabetes. But today I'm going to be really focusing just on the B cells. So B cells are able to recognize many of the beta cell proteins in type 1 diabetes. So these include GAD65, IA2, ZNT8, and insulin, and there's many others as well. And we know this is true because we can actually identify autoantibodies that are reactive to all of these different proteins. And what's really interesting about these autoantibodies is they're predictive of type 1 diabetes onset. So here I'm showing a plot where we have the incidence of type 1 diabetes on the y-axis and then the number of years on the x-axis. So one thing that you can see is that in individuals where no islet autoantibodies are detected, they have a very low probability of eventually developing type 1 diabetes. However, if there are 3 different islet autoantigens present, it is almost a certainty that within 20 years, this individual will develop type 1 diabetes. So these autoantibodies are really impressively predictive and currently one of the top tools used to predict type 1 diabetes. Additionally, we know B cells are important because there's several studies that have shown that B cell depletion is protective for type 1 diabetes. So here is a mouse study where in depleting these B cells in the mice, we actually see that those mice don't develop diabetes, while mice that have B cells present do still develop diabetes. And this is true both for male and female mice. And then additionally, in humans, there was a clinical trial done where they used rituximab, which again depletes B cells, and they found that those individuals treated with rituximab had a slower onset of type 1 diabetes compared to the controls. So today I'm going to tell 2 different stories. Both are going to be focused on B cells and diabetes. The first is looking at peripheral B cells, and then the second is trying to tie this into the pancreatic lymph node. So both stories are a collaboration with Dr. Mia Smith. This first story is a collaboration with Dr. Catherine Nicholas, who was a former student in Mia's lab. So in this study, we did single-cell RNA-seq of antigen-reactive B cells. So we have a cohort of non-diabetic individuals, autoantibody-positive individuals. So these are people where we detect autoantibodies, but they have not developed full-on type 1 diabetes yet. And then 5 new-onset type 1 diabetic individuals. And we use a very similar strategy to what Eric described previously, where we collect PMCs. We then have these antigen tetramers that have both a PE label and a barcoded tag. so that we can sequence out antigen-reactive cells, and then we can deconvolute them by sequencing in 10x. So we have insulin, I2, GAD, tetanus is our positive control, and then also empty, which is a DNA only. And what you can see is that we are able to identify these antigen-reactive cells by flow in all of our different cohorts. So we are able to sequence cells from each of these different individuals. The first thing we wanted to do was identify what B cell subsets were present, and we could see that we have naive, early memory, ABCs, memory, and plasmablasts. We see all expected B cell populations, and we do see all of these different classes across all of the different groups. So we don't really see a huge enrichment for any cell type in any specific group. Next, we want to really focus on what the reactivity status of these cells were. And here you can see everything in the pink colors are either positive or negative controls. And then the blue and green colors are islet autoantigens. And one thing that was really striking to us is we did see this expansion of B cells that recognized these islet autoantigens in autoantibody-positive individuals. And one thing that was especially interesting is we found a lot of these islet multi-reactive cells. So these are cases where the B cell was reacting to seemingly multiple of these islet autoantigens. So in this case, I'm showing here both insulin and GAD. We wanted to make sure that this was in fact a real result. So we decided to take and generate a lot of these different antibodies and validate them by ELISA. So at the top here, I'm showing places where we expect just one antigen to bind. And then between the two red lines are identified polyreactive cells. And then we tested them against all of the different antigens that we had in our panel. And you can see that largely we do see reactivity across many different antigens in our expected polyreactive cells. The one place where we don't is a case where we had a PE reactive cell. So this is actually something that Eric talked about using two different fluorophores, and that would help a situation like this. But other than this one situation, we do see largely polyreactive cells. And these islet reactive cells are actually identified in all populations. So here you can see all of our controls we see across all populations of B cells, and then this is also true for our islet multi-reactive and then each of our singly reactive antigens as well. When we look at these across the different groups, we see a similar pattern that I described before, where the autoantibody-positive individuals really have the most of these polyreactive cells. And of course, we don't see a lot of reactivity in the plasmablast, which is what you would expect because there's lower BCR. We next wanted to look at the gene expression patterns. And here we can see that there are really distinct gene expression patterns that tend to be more similar between the autoantibody-positive and type 1 diabetic donors. And when we start to really look at what gene sets are within these differentially expressed genes, we see a couple of pathways that come up. So here we did gene ontology enrichment, and then we clustered based on similarity of genes within the pathways. And we see that there's a lot of pro-inflammatory pathways. There's a lot of different infection pathways. These include bacterial, viral infections. We also have autoimmunity pathways, antigen pathways, and then also BCR and general signaling pathways. So a lot of these pathways tend to really point to a more activated immune system. Next, we want to really focus on the BCR because we did BCR sequencing as well. So we wanted to see if there was any enrichment of V gene usage in the autoantibody positive or type 1 diabetic individuals compared to control. So we just did a Fisher's exact test, and here you can see the odds ratio. Anytime when the odds ratio is high, this means that this V gene was seen more often in individuals with type 1 diabetes than in non-diabetic individuals. So if we look at those three V genes in autoantibody positive, we actually see the same enrichment. So now we can actually look at all V genes rather than just that small subset. And you can see that there are some cases where there's really great agreement between type 1 diabetes and autoantibody positive, and some cases where we only have enrichment in one of the two. But these are likely V genes that are more important for the development of disease. We also see some that have a reduction in type 1 diabetic individuals and autoantibody positive individuals. We can do the same thing now looking at both the heavy and light V genes rather than just the heavy alone. So instead of showing a huge plot with all pairwise possibilities, I'm now just showing only the pairs that we had enrichment for. This is a circus plot, so each individual line here is one cell where we saw that specific pairing. And right now I'm just coloring by the disease status. This is just to show you that our statistics work. You can see there are very, very few of these cells that are found in the non-diabetic individuals, and we've really seen enrichment for only autoantibody positive and type 1 diabetic individuals. If we take this same plot and we instead color it by the reactivity, we see that we are again really enriching for those islet reactive cells, so especially the blue polyreactive cells. So it seems like potentially these polyreactive cells are what are important in driving disease. We then wanted to look at clonal expansion. So again, using Immcantation to predict clonal expansion. And here we then again separated just based on the reactivity of those cells. So here we're showing all of cells that were within any clone, and you can see in the negative, empty, and tetramer, so all of our controls, we see a pretty even distribution across the different disease status groups. However, when we subset only for cells that were reactive to any of the islet antigens, we start to see a shift really away from the non-diabetic individuals and towards the autoantibody positive and type 1 diabetic individuals. And just to show this a different way, you can see that the clones in the non-diabetic individuals tend to really be either ones where they didn't bind to an antigen that was in our study, or they binded to tetanus. Where autoantibody positive individuals, we really see this expansion of polyreactive cells in those clones. We also are really interested in public clones. So these are clones that we see really similar sequences between different individuals, because we hypothesize that these shared sequences are going to be especially important in the development of disease. So here we're just looking at public clones, again, using a similar idea where we do heavy and light together rather than just heavy, and then we do 85% similarity of CDR3 for both heavy and light chains. And here you can see we had 31 clones that were found in 2 individuals, 45 that were found in 3, 6 that were found in 4, 4 that were found in 5, 1 that was found in 6, and 2 that were found in 7. And one thing that was really interesting here is that a lot of these cells, a lot of these clones are really enriched in the autoantibody positive and type 1 diabetic individuals, and we don't really see a lot of non-diabetic individuals that have these shared clones. So we hypothesize that these BCRs are more likely involved in the autoimmune response because we're now seeing them in multiple individuals rather than just in one. So just to summarize this part of the talk, we were able to find increased islet reactive and polyreactive B cells in type 1 diabetic and autoantibody positive individuals. We see changes in BCR signaling and autoimmunity pathways, and we also see changes in BCR repertoire. So now to go into the second part of the talk, this is again a collaboration with Dr. Mia Smith, but now this is working with Jessica Prendergast, who is a graduate student in both of our labs. And she's really looking at the clonal expansion in the pancreatic lymph node. So this is an in-progress project, and we're building our cohort of autoantibody positive type 1 diabetic individuals and also non-diabetic individuals. One big difference is, of course, the PLN can only be taken from deceased individuals. So this is a similar strategy, except we're not using antigen reactivity, so we're only doing surface markers, and then we're also doing single cell sequencing for both gene expression and BCR. So we're able to see the same populations that we saw in the PBMCs, which is really exciting. And again, we don't really see a lot of enrichment in either the autoantibody positive or type 1 diabetic individuals. And then the next thing we want to do is really look, are there any public clones that we see in both the PBMC and the PLN? And we actually do see cases where individuals where we see the same clone across many different individuals, with the brighter colors being our PLN samples and the darker colors being the PBMC samples. And again, you can really see that there's an enrichment for these autoantibody positive and type 1 diabetic individuals and not as many non-diabetic individuals in here. So when we start to really look at these shared clones, one thing that we see is that a lot of them are islet reactive. So if we just look at these 900 shared clones, you can see that, again, a lot of them have these islet autoantibodies, and we see the same pattern that we'd seen previously, where most of the autoantibody positive individuals have these polyreactive cells. If we just look at large clones, so clones with 10 or more cells, we see the exact same patterns. So again, we're really enriching for these islet reactive cells when we're looking at these clones that are also in the PLN. And one thing that's really interesting is that there are also clones that are skewed to one tissue. So we see this large expansion of clones in only one tissue. And this is particularly interesting if we look at something that's present in the blood, but then really becomes expanded in the pancreatic lymph node. When we again look only at clones that are in type 1 diabetic or autoantibody-positive individuals, we see the same pattern. And there are a couple of clones here that are probably particularly interesting where we see them in the blood, but we have this really large expansion of them within the pancreatic lymph node. So these are likely most interesting cells that you'd want to follow up on. So then just to summarize this part of the talk, we were able to identify public clones between B cells in the pancreatic lymph node and the PBMCs. These clones show islet reactivity, and there's also evidence of expansion of these disease-specific clones in the pancreatic lymph node. And with that, I would like to thank everyone who helped with this work, especially members of the Smith Lab, of course, all of the donors and their families, and all of our funding. And with that, I'm happy to take any questions.